Good evening, everyone. Thank you for coming. So my name is Parisa Khalili. I'm one of the medical directors in the solid tumor molecular oncology at ARUP and an assistant professor at the University of Utah. And the topic of my talk today is Introduction to Molecular Diagnostics. So it can be a really technical and a dry topic and subject to talk about, but I tried to simplify it and make it less technical, but still hopefully informative. So I have no disclosures to share with you today. This is the outline of my talk. First, I'm gonna go over some basics of molecular biology, just to serve as background information for the second part, where I'm gonna talk about 4 common molecular techniques that are used in clinical laboratories, and I'll discuss some of their advantages and limitations. So molecular diagnostic is actually application of molecular biology in patient care. And as you know, that the central dogma of molecular biology is that DNA is transcribed into RNA, and then RNA is translated into protein, which is associated with a phenotype. So any change in the DNA sequence can ultimately result in change in function or structure of the protein, which can be associated with a disease phenotype, such as cancer. Of course, you all know molecular testing has applications in different categories. In oncology, you can detect mutations in oncogenes or tumor suppressor genes, which give you information for classification, prognostication, or therapeutic decision making. For hereditary disorders, it will detect germline mutations in diseases of Mendelian inheritance, and of course has implications for patient and patient's family. And in the field of microbiology, by molecular testing, you can detect or quantify microorganisms, mainly viruses, and also identify genetic mechanisms of drug resistance. So tonight I'm not going to talk about hereditary disorders or molecular microbiology, and the focus of the talk is on field of oncology. So what is the genome? Genome is the entire complement of DNA in an organism. And in a human cell, genome is stored mainly in the nucleus, nuclear genome, but a small part of it is stored in mitochondria. So nuclear genome is basically made up of 3.3 billion base pairs. And these base pairs are, in a form of chromosomes, are spread unevenly. As you can see, they're like different size chromosomes in the nucleus. Just to a brief mention of mitochondrial genome, compared to the nuclear genome, it's very small. It only has 17,000 base pairs, and it's in the form of a circular double-stranded DNA. So what is in human genome? Of course, there are genes in the genome, and gene being a segment of DNA which has coding sequences. But turns out that very small amount of genome is made up of genes, maybe 1 to 2%, and a vast majority of the genome is composed of non-coding sequences, such as regulatory elements, repetitive sequences, or large duplications. So why do we care about human genome? Because cancer is a disease of the genome, as you know. And so if we look here at this schematic illustration of the genome depicted by the number of base pairs from 1 base pair to billions, There are a wide range of abnormalities or alterations that can happen in cancer, ranging from altered ploidy, where a cell has multiple copies of the entire of all the chromosomes, to gain and loss of one or part of a chromosome, structural abnormalities, small insertion deletions, and down to point mutations affecting only one base pair. So fortunately, we have different tools in a molecular diagnostic toolkit to use and detect such alterations based on the size and the nature of the abnormality. So I'm not going to talk about next generation sequencing. I just wanted to mention it briefly, that NGS is a very powerful technology, and can basically detect any kind of genomic alteration, any size or kind of alteration. However, it must be noted that NGS is a methodology and not a single assay. And clinical laboratories have different assays designed using this methodology for detection of different aberrations. And not all assays using NGS methodology can detect the whole range of these abnormalities. Okay, going from low resolution to high, let's talk about karyotype first. So karyotype is basically a whole evaluation of the whole genome from a chromosomal perspective. It uses a specialized lab technique that permits visualization of DNA in its bundled form. So here on the left, it is a picture of histone-depleted metaphase chromosome. It's an electromicrograph. So as you can see, there is a scaffold or core of a densely staining protein surrounded by a halo, and that halo is actually DNA. In contrast to the picture on the right, which shows the same metaphase chromosome bundled up around histones and other proteins. And here you can actually see the chromatid fibers here. Okay, so how is karyotype done? First, we get when the specimen comes in the cytogenetic laboratory, either blood or bone marrow or solid tumor, they're put in a tissue culture, and they are cultured in an incubator, 37 degrees, for 48 to 72 hours. And in order to ensure that we get enough number of metaphases, usually there's a mitogen, which is cell-specific, is added to this culture. And then when the cells are ready to be harvested, mitosis arrest is induced through addition of coulsamide. And then cells are harvested using hypotonic solution, which causes the cell to absorb water and become heavy and fragile, followed by fixation with methanol and acetic acid, which makes the cell more fragile, such that when you drop the cell suspension on a slide, you'll get a nice spread metaphase chromosomes. And then these chromosomes on the slide are then banded or stained with GIMSA for G-banding. And each chromosome has its own characteristic banding pattern. So based on these chromosomes are recognized based on that banding pattern and then assembled in order of size. And then the cytogeneticist can detect here not only numerical aberrations, but also through examining if the banding pattern is interrupted or not, detect if there's any structural abnormalities are present. As I said, you need to grow cells in an incubator. So you need to start with viable tissue and a tissue that can produce dividing cells, be it bone marrow or peripheral blood or fresh tissue. Therefore, it's important to put this specimen in a sterile fashion in a cytogenetic media and transport without delay to the cytogenetic lab. There shouldn't be any fixing or freezing. So solid tumors usually contain multiple chromosomal abnormalities and end up having a very complex karyotype. And so karyotyping of solid tumors is not routinely done anymore. However, I had an example, an older case, just to show you the benefit of karyotyping in solid tumors. This is a 10-day-old female with scalp lesion, and histomorphology shows bundles of malignant spindle cells. So the karyotype is done, and of course it shows a complex karyotype composition that consists of 5 related abnormal cell lines, which all of them have this translocation of short arm of chromosome 12 and long arm of chromosome 15, as well as copy number gain of chromosome 11. And it turns out that this translocation, 1215, results in fusion of ETV6 and NTRK3, which, in addition to copy number gain of chromosomes, is a recurring phenomena in infantile fibrosarcoma. There is another example of a 15-year-old male with a tibial lesion, and the requisition was to rule out osteosarcoma. And the karyotype was performed, and it showed a normal male karyotype. But it does not correlate with the morphology of a malignant tumor to have a normal karyotype. Therefore, so you need to remember that when cells are cultured, that is that is an artificial environment, and you have no control over what kind of cell is growing, and you don't know what is karyotype. So a negative study does not necessarily mean that genetic abnormalities are not present. So the key points, karyotype has some advantages. It provides an overview of the whole genome through evaluation of metaphase chromosomes, and it can detect both numerical and structural abnormalities. However, it's a very low resolution technique and can only detect large abnormalities in the range of 5 to 10 megabase. And it's dependent on proliferation in an artificial environment, which I just showed an example of that problem. And it's, of course, it's a long and labor-intensive procedure. So in order to overcome some of the limitations of karyotype, there's another technique can be used. It's fluorescence in situ hybridization, or FISH. And the concept of FISH is like detecting a needle in a haystack using a magnet. So you have a sequence of interest in the genome that you would like to evaluate, make a probe that is complementary to that sequence, and then label the probe fluorescently. And then so this way you can visualize your sequence of interest under the fluorescence microscope. FISH can be done on metaphase nucleus or on interphase. For the metaphase FISH, you'd need cell preparation like you'd need for a karyotype. But for so you cannot perform metaphase FISH on fixed or archival tissue. However, interface FISH can be done on a variety of specimen from FFP tissue or cell blocks to cytology preparations. And the sample adequacy for performing FISH is, depending on the probe strategy that I'm going to talk about later, is 50 to 100 intact tumor cells. But it must be noted that decalcification is something that will interfere with a yield of interpretable signals. And also certain fixatives and dyes, like the one that contain mercury or zinc, can also interfere with signal production. In general, FISH is the general FISH procedure is that you start with your specimen on a glass slide, and then DNA is denatured and hybridized overnight with your fluorescent probe. And then the next day, extra probe is washed, and the nucleus counterstain is added, which is called DAPI, and that's what gives the nucleus a gray-blue color. And then you perform analysis under fluorescent microscope. So as I alluded before, there are different strategies different probe strategies to detect different alterations. For copy number determination, enumeration probes are used, and for structural changes such as translocation, there are two kinds of probes that you can use, break-apart probes or dual-color, dual-fusion probes. So starting with enumeration probe, in this probe strategy, you have one probe targeting your gene of interest, in this example, HER2, on a long arm of chromosome 17, and then you have a control probe targeting the centromere of that chromosome, which in this case is centromere of chromosome 17 or CEP17. So a normal cell would show a signal pattern of 2 red, 2 green, meaning that 2 chromosomes 17 are present, and then you have on each one, you have one copy of HER2. So if you have multiple copies of one probe relative to the second probe, you can, based on if the number reaches the cutoffs, it can be a sign of amplification. So in this cell, for example, there are 2 green signals. So judged by the number of green signals, you have 2 chromosome 17, but you have multiple copies of HER2 signal. And so another scenario is when you have multiple copies of both the control probe and target probe, in this example, chromosome 12 and MDM2. So you can infer that what has happened is polysomia of chromosome 12, since although there are multiple copies, the ratio is still 1. One of the most important artifacts that needs to everyone who does the interpretation on solid tumor fish needs to keep in mind is truncation artifact. So as you know, in a paraffin block, an FFPE specimen, cells are not the cells are not in a monolayer. They're rather like overlapped on top of each other. And then when you take a section, like usually 5 micron section of that block, you'll only see a part of the nucleus, and sometimes they can show abnormal signal patterns. So there's also always like cutoffs set in the laboratory to account for this artifact, to call an assay result abnormal. So moving on to brachiport probes, in this strategy, you have a gene which is flanked by 2 different probes. The translocation breakpoint is flanked by 2 probes, and which are very close together in a normal situation. So when red and green are overlapped, you get a yellow signal. So a normal cell would show 2 yellow or 2 fusion signals. But then if there's a translocation, then these green and red signals will separate. So this is just like a kind of a classic abnormal signal pattern where you have 1 yellow, which is 1 fusion, and 1 red and 1 green, which shows that a rearrangement has happened and these 2 probes are separated. One thing to remember about these brachiport probes are that they can only give you information that if the rearrangement or translocation has happened in the cell, and they cannot give you information about excuse me, the partner chromosome. So there is a possibility of false negativity in this probe strategy. So as I mentioned, in a FFPE specimen, cells are not all in single plane. So for example, in this first picture, which is there is no translocation, you have 1 fusion, 1 red, 1 green, which there is a positive translocation. You have 1 fusion and then 1 separated. And then in the second image, there is no translocation. However, the green and red signals are overlapped, and you falsely get 2 yellow or 2 fusion signals, and might interpret that as negative. Okay, so the other probe technology is dual color, dual fusion, which is exactly the opposite of brachiport probe. So you have a probe for each gene on different chromosomes, and if in a normal situation you have 2 signals of each, 2 red, 2 green in this example. And if the translocation has occurred, they get close together, and then you get 2 fusion signals, 1 red and 1 green. And the advantage of this probe strategy is that it'll give you information about the partner chromosome in the translocation. And again, there is a problem of false positivity with this probe strategies. When there is a translocation, of course, you see separated red and green. But if they're overlapped, you see a fusion, and you might interpret that as positive. I just wanted to mention 2 kind of artifacts that we encounter with solid tumor fish. One is crush artifact. If you recall, for sample adequacy, I mentioned that we need 50 to 100 intact tumor cells. So this is an example of a small